Good day, StudyBuds! Welcome to UPTube. This is your StudyBud Joy, and I will be your companion in exploring the world of cell biology. Fasten your seatbelts and hold on tight, for it will be one whole marvelous of exciting trip. Let us define the cell first. The cell is the smallest unit that can carry on all of the processes of life. All living things are made up of cells. But how do we know that living things are made up of cells? And who discovered cells? Do you know Robert Hooke? He is an English scientist who studied nature by using an early light microscope. He looked at a thin slice of cork from the bark of a cork oak tree and described great many little boxes that reminded him of a cubicle or cells where monks lived. He indicated that he had clearly observed the remains of cork cells. How about Anton van Leeuwenhoek? Do you know him? He was the first person to observe living cells. In 1673, he was able to observe a previously unseen world of microorganisms. He called those organisms animalcules. We call them now protists. Matthias Schlitten. He was a German botanist who concluded that all plants were composed of cells. Theodor Schwann was a German zoologist who concluded that all animals were composed of cells. And also Rudolf Virchow, a German physician who noted that all cells come from other cells as he studies human diseases. The discovery of cells serves as an observation. They were combined to form a basic theory about the cellular nature of life. The cell theory has three essential parts, which are summarized as: first, living organisms are composed of one or more cells. Second, cells are the basic unit of structure and function in an organism. And last, cells come only from the reproduction of the existing cells. Let us pack our things and be ready for our departure as we finish the history of cells. Let us now explore the cell structures and their functions. The plasma membrane or the cell membrane is the cell's outer boundary. It covers the cell surface and acts as a barrier between the outside and the inside of a cell. All material enter or exit through the plasma membrane. The cytoplasm is a region of the cell that is within the plasma membrane and that includes the fluid, the cytoskeleton, and all of the organelles except the nucleus. There is a part in the cytoplasm that is called cytosol. It includes molecules and small particles such as ribosomes but not membrane-bound organelles. About 20% of the cytosol is made up of protein. The nucleus, also called the control center of a cell. Most of the functions of a eukaryotic cell are controlled by the cell's nucleus. The nucleus is often the most prominent structure within a eukaryotic cell. With the help of a protein skeleton called nuclear matrix, it can maintain its shape. There are two basic types of cells. First, the prokaryotes. They are organisms that lack a membrane-bound nucleus and membrane-bound organelles. Prokaryotes are divided into two domains, bacteria and archaea. The domain bacteria includes organisms that are similar to the first cellular life forms. The domain archaea includes organisms that are thought to be more closely related to eukaryotic cells found in all other kingdoms of life. Second, the eukaryotes. They are organisms that are made up of one or more cells that have nucleus and membrane-bound organelles. Eukaryotic cells also have a variety of subcellular structures called organelles, well-defined intracellular bodies that perform specific functions for the cell. The organelles carry out a cellular processes just as a person's pancreas, heart, and other organs carry out a person's life processes. Let us now proceed to the cell organelles and their features. First, the plasma membrane. The plasma membrane, also called the cell membrane, allows only certain molecules to enter or leave the cell. It separates internal metabolic reactions from the external environment. Plus, the plasma membrane allows the cell to excrete waste and to interact with its environment. The plasma membrane has three subparts: membrane lipids, membrane proteins, and fluid mosaic model. Membrane lipids The plasma membrane, as well as the membranes of cell organelles, is made primarily of phospholipids. Phospholipids have a polar hydrophilic phosphate head and two non-polar hydrophobic fatty acid tails. Water molecules surround the plasma membrane. The cell membranes of eukaryotes also contain lipids called sterols between the tails of the phospholipids. The major membrane sterol in animal cells is cholesterol. Sterols in the plasma membrane make the membrane more firm and prevent the membrane from fizzing at low temperatures. Membrane proteins. Plasma membranes often contain specific proteins embedded within the lipid bilayer. These proteins are called integral proteins. Some integral proteins, such as cell surface markers, emerge from only one side of the membrane. Others, such as receptor proteins and transport proteins, extend across the plasma membrane and are exposed to both the cell's interior and exterior environments. Proteins that extend across the plasma membrane are able to detect environmental signals and transmit them to the inside of the cell. Integral proteins play important roles in actively transporting molecules into the cell. Some act as a channel or pores that allow certain substances to pass. Other integral proteins bind to a molecule on the outside of the cell and then transport it through the membrane. Still others act as a site where chemical messengers such as hormones can attach. The fluid mosaic model. Scientists describe the cell membrane as a fluid mosaic. The fluid mosaic model states that the phospholipid bilayer behaves like a fluid more than it behaves like a solid. The membrane's lipids and proteins can move laterally within the bilayer like a boat on the ocean. As a result of such lateral movement, the pattern or mosaic of the lipids and proteins in the cell membrane constantly changes. Second, the nucleus. Most of the functions of a eukaryotic cell are controlled by the nucleus. The nucleus is filled with a jelly-like liquid called the nucleoplasm, which holds the contents of the nucleus and is similar in function to a cell cytoplasm. The nucleus houses and protects the cell's genetic information. The hereditary information that contains the instructions for the structure and function of the organism is coded in the organism's DNA, which is contained in the nucleus. The nucleus also has subparts: nuclear envelope and nucleolus. Nuclear envelope. The nucleus is surrounded by a double membrane called the nuclear envelope. The nuclear envelope is made up of two phospholipid bilayers. Covering the surface of the nuclear envelope are tiny protein-lined holes, which are nuclear pores. The nuclear pores provide passageway for RNA and other materials to enter and leave the nucleus. Nucleolus. Most nuclei contain at least one denser area called the nucleolus. The nucleolus, or plural, nucleoli, is the site where DNA is concentrated when it is in the process of making ribosomal RNA. Ribosomes are organelles made of protein and RNA that direct protein synthesis in the cytoplasm. Third, mitochondria. Mitochondria, or singular mitochondrion, are tiny organelles that transfer energy from the organic molecules to adenosine triphosphate, or ATP. ATP ultimately powers most of the cell's chemical reactions. Similar to nucleus, mitochondrion has an inner and an outer phospholipid membrane. The outer membrane separates the mitochondrion from the cytosol. The inner membrane has many folds called cristae. Cristae contains proteins that carry out energy-harvesting chemical reactions. Fourth, ribosomes. Ribosomes are small, roughly spherical organelles that are responsible for building protein. Ribosomes do not have a membrane. They are made of protein and RNA molecules. Fifth, endoplasmic reticulum. The endoplasmic reticulum is a system of membranous tubes and sacs called cisternae. The ER functions primarily as an intracellular highway, a path along which molecules move from one part of the cell to another. The amount of ER inside a cell fluctuates depending on the cell's activity. There are two types of ER, rough and smooth. The rough endoplasmic reticulum is a system of interconnected, flattened sacs covered with ribosomes. The rough ER produces phospholipids and proteins. Certain types of proteins are made on the rough ER's ribosomes. These proteins are later exported from the cell or inserted into one of the cell's own membranes. Rough ER is most abundant in cells that produce large amount of protein for export, such as cells in digestive glands and antibody-producing cells. The smooth ER lacks ribosomes and thus has a smooth appearance. Smooth ER builds lipids such as cholesterol. In the ovaries and testes, smooth ER produces the steroid hormones estrogen and testosterone. In skeletal and heart muscle cells, smooth ER releases calcium which stimulates contraction. Smooth ER is also abundant in liver and kidney cells where it helps detoxify drugs and poisons. 6. Golgi apparatus. The Golgi apparatus is another system of flattened membranous sacs. The sacs nearest the nucleus receive vesicles from the ER containing newly made proteins or lipids. Vesicles travel from one part of the Golgi apparatus to the next and transport substances as they go. The stuck membranes modify the vesicle contents as they move along. The proteins get address labels that direct them to various other parts of the cell. During this modification, the Golgi apparatus can add carbohydrate labels to proteins or alter new lipid in various ways. 7th. Vesicles. Vesicles are small, spherically shaped sacs that are surrounded by a single membrane and that are classified by their contents. Vesicles often migrate to and merge with the plasma membrane. As they do, they release their contents to the outside of the cell. There are different types of vesicles. First are the lysosomes. Lysosomes are vesicles that bud from the Golgi apparatus that contain digestive enzymes. These enzymes can break down large molecules such as proteins, nucleic acid, carbohydrates, and phospholipids. Lysosomes are also responsible for breaking down cells when it is time for the cells to die. They play a very important role in maintaining an organism's health by destroying cells that are no longer functioning properly. The peroxisomes. Peroxisomes are similar to lysosomes but contain different enzymes and are not produced by the Golgi apparatus. They break down fatty acids, which the mitochondria can then use as an energy source. There are also other vesicles. Specialized peroxisomes called galactosomes can be found in the seeds of some plants. They break down stored fats to provide energy for the developing plant embryo. Some cells engulf material by surrounding it with plasma membrane. The resulting pocket pods often become a vesicle inside the cell. This vesicle is called an endosome. Lysosomes fuse with endosomes and digest the engulfed material. Food vacuoles are vesicles that store nutrients for a cell. Contractile vacuoles are vesicles that contract and dispose of excess water inside a cell. 8. Cytoskeleton. The cytoskeleton is a network of thin tubes and filaments that crisscrosses the cytosol. The tubes and filaments give shape to the cell from the inside, in the same way that tent poles support the shape of a tent. The cytoskeleton also acts as a system of internal tracks on which items move around the cell. Cytoskeleton's functions are based on several structural elements. 3 of these are microtubules, microfilaments, and intermediate filaments. Microtubules are hollow tubes made of a protein called tubulin. Microtubules hold organelles in place, maintains a cell shape, and act as tracks that guide organelles and molecules as they move along within the cell. Microfilaments are long threads of the bead-like protein actin and are linked end-to-end and wrap around each other like 2 strands of a rope. Microfilaments contribute to cell movement, including the crawling of white blood cells and the contraction of muscle cells. Intermediate filaments are rods that anchor nucleus and some other organelles to their places in the cell. They maintain the internal shape of the nucleus. 9. Cilia and flagella. Cilia and flagella are hair-like structures that extend from the surface of the cell, where they assist in the movement. Cilia are short and are present in large numbers on certain cells, whereas flagella are longer and are far less numerous on the cells where they occur. Lastly, the centrioles. Centrioles consist of 2 short cylinder microtubules at right angles to each other and are situated in the cytoplasm near the nuclear envelope. Centrioles occur in animal cells, where they organize the microtubules of the cytoskeleton during the cell division. Please note that plant cells lack centrioles. That's it for today's adventure, intellectual learners. Let us remember that the cell is the natural granule of life. In order to understand life, we must try to understand the cell. This has been your StudyBud Joy. Till the next trip, StudyBuds!